This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 528, recorded on January 4th, 2019. Happy New Year, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me here in a gray and cold New York City, Dickson, Despommier.
Oh, Vincent.
It's sort of looking like winter, right?
It started out good this morning, but it doesn't look so great right now.
Yeah,
but it's still warmish.
No, it's not. It's freezing.
No, it's warmish.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it's 44 and mostly cloudy here.
Yeah, that's warmish.
It's warmish here this morning.
44.
Frozen. You know, I look out the window, I look at the Pachysandra. When they're all shriveled up, it's frozen out. Do you know what I'm talking about?
Oh, I look at the rhododendrons.
Yeah, I don't have any roadies. No roadies.
Rhododendron leaves roll up when it's below freezing.
Yeah, it's like,
roll up tighter the colder
listen,
it gets.
if you guys were where I was last week in Portugal, the flowers were blooming. All of them. The hibiscus, everything was out. They said it's all like this all year long. I said, oh, my goodness.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody.
Hey, Rich.
We have, finally, after days of cold and gray, we have 62 Fahrenheit, 17 Celsius, and bright blue sunny skies. It's gorgeous.
Nice.
Cool.
And from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. We also have bright blue sunny skies, not a single cloud, 42 Fahrenheit, 6 Celsius.
Goodness.
When I was in Wisconsin, I spoke with a plant biologist who they work on a...
I thought you were just going to say I spoke with a plant.
I've been known to speak with plants, yeah. He works on a plant that... How does this work? It has to go through like a winter before it will flower again. Temperature
I think
based.
a lot of them are like that.
Yeah, so there's some which require a cold for extended period of time before they will flower. And he's working on it. It's all transcriptional regulation and thermosensing. It's really cool.
Yeah. Mm-hmm.
I know apple trees, the yield is dependent on how bad the winter was, and really, really bad winters apparently lead to better apple crops.
Yeah.
Apple Trees LLC, right? Isn't that your company?
That's what my last name means.
Apple Trees.
Apple Trees.
Now,
as long as we're doing the weather on a year-end show, I just have to say that on the 1st of January, I noticed the weather report in the paper, and it included some cumulative annual data. So in Austin, we had the normal rainfall, this is important to me, is about 35 inches, little less. And we had, depending on where you measure it, there's two different sites where they take data, between 4 and 18% more than that. So we're fixed good for rain, and all the reservoirs are full.
Yeah, that's unusual.
Awesome.
Very good.
Yeah, that's an unusual thing
great.
for Texas. That's
I like that.
You're also fixed for mosquitoes.
The high temperature for the year was 110.
Exactly.
The low temperature for the year was 15, which happened on this day a year ago.
All right.
And the other notable bit of data is that there were, depending on where you count them, between 41 and 52 days that were in excess of 100 degrees.
Right.
Whoa.
Right.
Yeah. Whoa.
So not only do I think of West Nile when I think of that, but I also think about the citrus industry. And I love oranges and grapefruits in the morning for breakfast. I mean, one of each, of course. They have been so difficult to get, good ones. Florida's had bad weather, California's had fires, and Texas has had all this drought. And the citrus industry has really suffered for this.
Mm-hmm.
Well, it's coming back.
Right.
Dickson, Texas is going to send you some grapefruits next week.
Would they? Oh, I would just be thrilled. Trilled.
Citrus tristeza.
Citrus. In fact, they used to call the baseball league down here the citrus circuit. The citrus league is,
Yeah, citrus circuit.
Exactly.
This Week in Virology wants to learn more about you, the listeners of TWiV. Please take 5 minutes to answer a short 12-question survey. This will help us collect data needed to seek sponsorship and make the podcast I don't know, the word here is grow, but I don't like you to grow.
No, it's already 2 hours long.
How about mature? Mature
the podcast. You can find the poll at asm.org/TWiV poll, asm.org/TWiVPOLL, one word. And while you're at it, you could go to TWIM poll and you'll get the same thing for TWIM. Thanks for your time and listening, subscribing, and helping us improve the show. We appreciate your support.
How many people have done this so far, Vincent?
I don't know. I don't have access to the stats, but let me see. I can get them in real time. As Dickson, were you going to say something?
Yeah, I was going to ask you if we could get this for TWiP also.
Yeah, we could.
Good.
I'll ask Ray right now. I'm going to text him. Today is Ray Ortega's birthday, by the way.
Hey,
Happy birthday.
And he doesn't listen to TWiV probably. Even though he posts it at asm.org. I love this society, this world we live in where you can contact people. In my day, I had to put dimes in telephone poles.
In my day, I had to light a fire and get a rug.
So while you're texting him, text him happy birthday from the TWiV crew.
Absolutely.
Happy complete from the TWiV, TWiV crew. Of course, the problem with these technologies is spell checking, which I dislike very much. Yeah. Because they always change words that we use that are not in the dictionary, right?
Yeah, but what's interesting too is that they learn, like I'll bet you when you put in TWiV, it did all the
TWiV is good.
Caps and lowercase, yeah.
But I've been texting neurotropism lately, okay, and it hasn't learned that yet.
It's a bigger word. Come on.
Come on, learn neurotropism. Kathy, any news about ASV?
Oh, pretty much the same as always, except that I checked the stats on dues renewals, and they're really poor. So a lot of people must be procrastinating, but they shouldn't do it any longer because abstracts are due February 1st. Sponsors need to be current with their dues paid. Students and postdocs who want to apply for travel grants need to be members. And if you want to apply for one of the ASV Cares Dependent Care Fellowships, you need to be a current member. So pay your dues. Yikes.
I did it I did it live on Twitter last
time. Yes, you did. Yes.
It worked.
So, yes.
Okay. I want to announce the European Congress of Virology meeting in 2019. This is happening in Rotterdam, Netherlands.
It's the world's largest port city.
Is that right?
Yep.
I'm looking at a picture on the website, ecv2019.com. A lot of big buildings there.
Right.
So this is the reason I'm telling you this is because it's being run by, the Marion Koopmans and Ben Berkhout. And they have asked me to go and do a couple of TWiVs, actually two. Two TWiVs. Wow. That's the first time, too. But it's also a cool meeting. It's, the European Congress of Virology.
Sure.
It's a big organization.
What month is that? I'm sorry.
End of April.
End of April. Okay.
So April
and there's if you want to go, there's an abstract deadline of March 1st, 2019.
So that'll be right around the time the tulips are blooming, right?
Yes.
No idea.
No. Ellen's right. That is.
Cool.
Looks like an interesting city.
And by the way, they have the world's largest flower auction there as well.
Mm-hmm.
And there's a building that's huge, absolutely huge. And all of the flowers to the world from Holland are sent via that building. And they have the most amazing sorting systems, and it's automated. And it's really quite amazing, actually.
So Marion Koopmans is head of virology at Erasmus Medical Center. She's awesome. She was on a TWiV that we did at Boston University. Ellen was there.
Yeah.
And she's great because she just chimes in on everything and really knows her stuff. And after it, she goes, this is one of the most fun things I've done in a long time. We have to do this when I have this meeting in Rotterdam. So that's why it's happening.
Good. Yeah, she's really smart, really insightful.
Lovely.
So the Holland official guide for visiting Holland says that tulips are mid-April through the first week of May.
Perfect.
This is a perfect timing.
Maybe I could see some tulip break.
When
Maybe
you
that's
land,
why
maybe.
No, wait a minute. When you land at Schiphol Airport in Amsterdam, you will see tulips growing on the grass between the runways.
Is that right?
They don't lose any ground for that. They make use of every inch.
I have a tulip story. Years ago, Peter Palese went to Holland. He always brought his wife some present, and he brought tulip bulbs.
Yeah.
And I remember he took me to his house one night for some reason, and he had planted them, and there was one here, one there on his front lawn, and they were just sticking up. He said, I didn't know what to do, so I put one here and one there. It's no good.
Eventually, you'll have a lawn full of tulips because
really
they do
funny.
spread.
It was really funny. Tulips are really nice.
They're great flowers. Yeah, they are wonderful.
You can't bend them, though.
No.
They snap.
No, they do.
I want to tell you again about our AFM crowdfunding effort. We are at 62% of our goal of $15,000. And I want to shout out Charlene Rayside in memory of William T.G. Jr., James Kiru, Erica Sapphire, Syra Loftus in honor of Orville Young, Alex Loop, Shrijun Zhang, Joseph Rabinowitz, Judy Becker in honor of Margot Becker, Margaret Helminska in honor of Julia Rose Payne, Joseph Kurland, Mark Martin in honor of Abigail Salyers and Edward Ledbetter, Jonathan Deloach, Pamela O'Neill, Sung Sil Moon, Paula Amis, and Sinew Huang. So Mark Martin is a microbiologist who was on TWiV, and these are, he said, his two heroes of microbiology.
And Erica Sapphire, is that Erica Ullman Sapphire?
It is. Scripps.
Okay. Some Scripps.
Yes. So thank you. And there are also two of my neighbors here who donated. Thank you very much. We had some virology birthdays this week. So mine was the 2nd of January, and I'm now Route 66.
You are.
Rich, do you remember when you were Route 66?
No, I remember when I was 64 because I had the Beatles song when I was 64 going through my head the whole time.
Right.
So 64, I think, in base 10 is like one with 6 zeros, right?
That beats me.
Yeah, so that was cool that year. That was amazing.
Yeah,
something like that.
Let's see. It's in hexadecimal. It's one of them you're 40, and one of them you're. I knew it for last year, but it's too late now.
Well, the way I think about it is this: you don't pay attention until you get middle age, and then you hit 55. That's the speed limit now, right? So the next year that you hit like that is 66. So now you can go 55 miles an hour on Route 66. And then the next one you hit is 77, which I hit last year. So that's 77 Sunset Strip. You can actually do 55 miles an hour on 77 Sunset Strip, but you'll get arrested. And finally, when you get to be 88, you can play the piano at your own funeral.
Well, you're morbid.
Well, 88's a reach for most people. That's not the average lifespan.
In some states, the speed limit's 75.
I know. So that
it's not
you'll be playing your piano at a younger age in those states.
Yeah. It's not a double number, yeah.
I understand. I was just in Wisconsin. And
I have nothing for 99, by the way.
So the virology birthdays, besides me, Paul Benash, I believe, was also January 2nd. Harmeet Malik was yesterday, January 3rd, and today, Ray Ortega. And there are probably many more, but those are the ones I know offhand. And it's an interesting time to have a birthday at the beginning of January. Our sons has a birthday on Sunday, the 6th. In fact, when we were waiting for him to be born, I said he's going to be born on the 2nd, but nope, didn't happen. So
they never do that on time.
Could have arranged it.
This is our summary episode of last year, all the cool things. And we released 51 episodes in 2018 and 2 special episodes. Peter Hotez and David Tuller. And Dickson, you missed Peter Hotez.
Yeah,
he was here briefly.
Yep.
We did 14 road shows.
That's a lot of road shows.
We did.
Wasn't Raul's an extra one too?
Now, his was a normal one.
He's got a number?
He's got a number.
Okay.
Yeah, they released it midweek, but I put a number on it.
Okay.
I want to get to 1,000 more quickly, you know?
So we've had numberless guests on the show.
Right.
We had 14 road shows, and we released 8 in a row this summer from the road. 11 Skype or in-studio guests. That's down from 14 the previous years, and we did 13 episodes with video. And the downloads were pretty consistent. They ranged from a low of 43,777 to 57,000. And that's all episodes, not just the ones released in 2018, because people always go back to the older episodes and listen. And they're there forever. So they contribute. I mean, they contribute less than the ones that are released.
By the way, do you think they're all out near the Kuiper Belt right now? Because we just had a remarkable event out there.
Yeah.
Well, you could actually calculate that, right? How long has the show been going on, and then just light years distance out?
Right.
I think we're probably out somewhere past Alpha Centauri at this point, aren't we?
Yeah. It only takes 6 hours to talk to that spacecraft out in the.
But that assumes that our podcast is actually being transmitted on some frequency that could that could travel there, and I don't think we're on any radio stations.
No, we're not.
Too bad.
This is podcasting. It's different.
Yeah, but the aliens all listen to podcasts.
They do, especially the cooking shows.
I mean, I guess we go over Wi-Fi.
Yep.
But that's not.
I got to say, speaking of aliens, I saw what is it called, Arrival this week.
Yeah.
I'd never seen it, and that is so cool. The concept of the aliens and how they communicate. It was so cool. Right?
It was very good.
Right.
And I really liked the idea of using a language professor to talk. That was just brilliant. Yep. Although it fell apart at the end. It totally fell apart.
You're quite right.
But
I don't know. I kind of like the ending.
Getting the phone call from the future, that's ridiculous. That's just so
I thought that was a pretty cool twist.
Tacky. And then all of a sudden she marries this guy.
By the way, spoiler alert.
She marries this guy who she no chemistry with whatsoever throughout the whole movie. And all of a sudden they have a kid. That's it.
Yeah, that was a little lame.
Anyway, I think the concept of the aliens, when you think, what would an alien, it doesn't have to be humomorph. And they're cool. They're kind of nebulous. And I like their names. What are they? Pentapods, they called. Or octopods? I don't remember how many legs they had. It's total downloads for 2018, 610,000 and change.
Which averages out to 50,859, which means that June and January have close to the average number of downloads.
Oh, yeah.
Oh no, wait. Not January. Sorry.
No, not January.
No, June. June has very close to the average number.
That's a lot of downloads. So it's really a well listened. It's more than
in October
any of our other shows, but that's because we're weekly and we've been doing it for 10 years, and you amass an audience. And we started,
and people seem to download pretty promptly. I was interested to see that January 55,000 downloads and change, and December 52,000 downloads and change. So people are downloading within the month for the most part, and that's certainly good.
All-time TWiV downloads are 5.8 million plus.
Cool.
So very cool.
How do we estimate our listener base?
You have to look at downloads.
Well, I mean, how many people do you think that averages?
Well, I don't know, because some people may download twice, right?
Yeah, sure.
I just don't know.
Right.
What do we have for subscribers? Like 20,000 plus?
I just don't know anymore. It used to be.
Because they're on different platforms.
So now they're not just iTunes anymore.
Right.
They're on all different. So we are on, what's the latest one?
Probably the way to.
So Stitcher Radio we are on. And
Overcast.
And Pandora just started. They just started podcasts, and they asked TWiV to join, which was cool. They're on beta now, I believe. And then the other one, what's the other music sharing?
iTunes?
No.
Spotify?
I think we're on Spotify as well.
Okay.
But all those numbers come through here.
I see.
If you're interested, the most downloaded episode in 2018 was 475, the first episode of the year, 10,228. That's the year summary of 2017. So suppose
people make a New Year's resolution to listen to more virology.
Right.
I don't know.
Yeah, exactly. And then they fall off the wagon.
Or just catch up on the whole year by listening to that one episode.
Yeah, they just listen to one.
That's right.
Or they get a new iPhone or something, and they want to listen.
That's right. They take a couple of swigs of Tamiflu, and we all go away.
Of course, last year we did number 500, which was pretty cool.
That's one of my
favorites.
favorites.
It was a great trip to Austin, Texas, and
it's fabulous.
Yeah. Thanks to Chris Sullivan again for making that all happen.
We all went.
Yeah.
Yes.
So it's rare that we're all in one place, right? And it was
I remember?
It was a good show. I also want to thank some people who have helped us. Jolene for doing the timestamps. She does them not only for TWiV, but for TWiVo and Immune.
Jolene is awesome.
And she arranged a trip to Texas A&M while we were in Texas last summer. So that was very cool.
Sorry, I missed that one.
Yes, it was fun. It was hot, but I loved it.
Yep.
I want to thank ASM, the American Society for Microbiology, and the American Society for Virology for supporting live episodes. We did a TWiV at each of those meetings. And I also want to thank the universities and meetings who hosted TWiV last year. That includes the University of Zurich, Penn State Hershey, the Double-Stranded RNA Symposium in Belgium, the retroviruses at Cold Spring Harbor, which Dickson
That was
went to.
that was lovely.
Took us 4 hours to drive there from Manhattan. Ridiculous. Texas A&M, University of Texas, Austin, the Smithsonian Institute, Outbreak,
Right.
Canadian Society for Virology in Halifax, where Alan went as well.
Yep.
Europic 2018, that was in the Netherlands. University of California, Irvine, where Rich and I did a vaccine symposium. Drexel University and the Medical College of Wisconsin. TWiV was peripatetic, you would say, Dickson?
Yeah, absolutely.
So to recognize this extensive travel, Sharon Isern designed us a special edition TWiV World Tour 2018 T-shirt. And you can buy, it's available, excuse me, it's available now on Amazon.
Really?
Yes.
Oh, cool.
So I put a link in the show notes. You can, she has a store, steampunkphage.com.
It's amazing.
And she makes lots of designs and puts them
I
on
had no
t-shirts.
idea she had this talent.
Yeah.
It's really cool.
And so you can buy them on Amazon. They're black, and there's a front which has a nice picture that she made, and then the back is a list of all the world tour sites, just like a rock T-shirt.
Outstanding.
This
The
is awesome.
the steampunk phage is awesome.
Yeah,
I can get that as a T-shirt.
Yeah. Anything on her site they're all T-shirt designs. If you go to that gallery, you click one that you want, and then at the bottom it says buy at Amazon or available here.
Cool.
And it's neat. I have the steampunk one is very cool. Steampunk phage. And there's one that Zika in the Americas, which I really like. It's a mosquito carrying Zika to the Americas, which is full of mosquitoes.
Oh, that is really good.
I have that one too. Phage versus bacteria. Yeah, she has a great art style. I like it.
Yeah, I like it a
This
lot.
is cool.
So anyway, get your TWiV World Tour 2018 T-shirts. What size do you need, Dickson?
Extra large. Not for long, however. I'm planning on gaining a lot more weight. No, that isn't true. We're trying to cut back on our calories so that, every New Year's.
I've heard that.
Yeah, me too. It's a well-worn statement.
Now, let's go through about 10-ish cool stories from Virology 2018 in no particular order. And I'll just start with one story that really intrigued me this year, in which I really had not been aware of. That is the potential involvement of viruses in Alzheimer's disease. And we did 2 episodes, TWiV 505, which was called Rose Ebola, and TWiV 519, Fishing for Viruses in Senile. And, you cannot tell often what the episodes are about by the titles, but at least they're fun.
You can't.
I would say usually.
Usually you
That's right.
can't.
But the title should make you want to know what the episode is.
Some people get mad and say, I can't tell what's in the episode. Well, why don't you just listen to it?
Or just, if you,
Just click
as soon
on
as you
the
go to the
notes.
episode page or
That's
the
right.
page on iTunes or whatever, you find the
There's a
subtitle of it and the little description of the show that's in there, and that tells you what's in the show.
That's exactly.
So Rose Ebola, TWiV 505, that was in August, and that's when Alan picked Impossible Burger. This is big.
I was trying to remember the name of that thing the other day. Yeah.
There you go. So this was the first time we did this paper, and this was, no, it's not the first time. Is it 505? I guess so, because the other one was fishing. Yeah, this is a paper in Neuron from Mount Sinai where they had looked in archived brain specimens from Alzheimer's patients and also normal controls, and just found high levels of herpes viral transcripts in the AD brains compared to the normal ones. And apparently this has had a long history of investigation, herpes viruses in the CNS association with Alzheimer's. In the same episode, we talked about how beta amyloid production is a response to infection and may be an innate response to infection. Of course, that's characteristic, the deposition of beta amyloid. Then later, TWiV 519, we revisited it because we had expressed skepticism at that first August episode. We revisited it and went more deeply into some of these stories. And at the time, I learned that some clinical trials were enrolling to test the efficacy of a herpes antiviral in preventing the progression of Alzheimer's, shall we say. And I found 2 actual clinical trials. Everyone probably knows clinicaltrials.gov is a great way to look for NIH-funded clinical trials. You can search for keywords. So if you look for Alzheimer's and herpes virus, you will find one that's enrolling here at Columbia. And the summary is antiviral therapy in Alzheimer's will investigate the efficacy of treating patients with mild Alzheimer's with the USA-marketed generic antiviral Valtrex, valacyclovir, 2 grams to 4 grams a day for 18
approximately
months.
1 shovelful.
Exactly.
It's a lot. So they're going to have
Half a cup.
130 mild patients, and
Goodness.
half of them will get drug and half will get a placebo. 18 months. So they do a PET scan at the onset, and then they do a PET scan at the end to see if it's prevented any advancement. Tau PET imaging at baseline and at 78 weeks. So it's still enrolling. So we're not going to have this for a while. But this is going to be very interesting. And we had skepticism because we don't think there's actually lytic virus replication in the brain, at least herpes virus, because you'd be in deep trouble. But there can be abortive replication. And maybe if you have the thymidine kinase gene expressed, you would get a target for valcyclovir, which might do something else. Who knows?
Yeah, so I think I'm certainly still skeptical, but interested.
I'm very interested. And the other clinical trial is taking place at Umeå University. I guess that's in Sweden, right?
Yes, it's in northern Sweden.
Umeå. Again, valcyclovir treat Alzheimer's patients. Open trial, 36 participants will receive only 4 weeks of treatment. It's interesting. So it's much shorter. And this is also enrolling at the moment. So these are NIH-supported clinical trials. I'm really interested to see what happens there. And I'm sure we're going to see more papers in the next year on this as well. So this one really stood out for me because, boom, it came out of nowhere. And there's a lot of back history to this. So that was one of the cool stories that I thought might be interesting. Another one was really stunning to me, TWiV 506, which was called A Cafeteria Full of Jelly Rolls. Apparent elimination of dengue in an Australian city by release of mosquitoes engineered to have Wolbachia. Aedes aegypti.
Yeah, this is cool. I'd forgotten about that.
I mean, Aedes aegypti, Wolbachia is present in many insects globally, but Aedes aegypti not. But if you put Wolbachia in endosymbiotic bacteria, it will inhibit the replication of a number of viruses, including
and some protozoa.
Dengue. Yes, which ones?
Malaria.
Yep. That's a good strategy for malaria treatment.
Yeah,
prevention?
but Anopheles is the vector, not dengue.
You'd have to do it in Anopheles.
Yeah, that's right.
Not Aedes.
No.
Okay.
That's a culicine mosquito.
Mm-hmm. And it's a question of the angle of standing on this.
Exactly.
Dickson taught me that years ago. Anyway, I think this is really amazing. So I found a cool website, which is called the World Mosquito Program. And they have, and there's a map of all of their releases that they have planned in the future. If you go to projects from their project sites around the world, there's Mexico, Colombia, Brazil, and a bunch near Australia. In Australia, we've talked about Indonesia, Vietnam, India, Sri Lanka. And so they are trying to get low-cost release programs in place.
Right.
I know they don't want it to cost a lot of money. And I think this is awesome. They have a nice summary of how it works and so forth there as well.
Right. The beauty of using Aedes is that you can ship the eggs because they can dry out and they'll still be viable. So you can treat them like they're letters
They say we are currently adapting our approach for use in large urban environments and targeting a cost of US $1 per person. So this could cost, what, $7 billion for the whole world, right?
That's worth it. You know how much dengue costs the world?
you don't have to do the whole world. You only have to hit the dengue zones, but that is going to be that's going to be a few billion people.
Exactly.
So I guess you would not do this in the U.S., right?
No, we don't have much dengue in the U.S.
I don't think it'd be worth it.
Not even a lot of Aedes aegypti either. We have Aedes albopictus instead.
Well, we have Aedes aegypti.
Some, not much.
Well, if you look at the map, there's
no, but it just says the states where they found it. There isn't a lot there.
But we also have ubiquitous window screens and air conditioners in places that do have Aedes aegypti, and that's another factor.
Yeah. So anyway, very interested to see where this goes.
That's true.
We'll hear about it, I'm sure.
Yeah,
so a really huge effort.
It is.
It's interesting if you click in the projects thing on this website. If you click on the site in Australia, there's a picture of a little kid.
Yes.
In his backyard with a thing that's called the Mozzie box.
Yes.
Yep.
Mozzie, I guess, is Aussie for
that's right.
Mosquitoes, right?
The
and I guess that's he probably got mosquitoes in his Mozzie box to release, I guess.
Yeah, they involve the kids in the schools. That is very cool.
A critical part of this, and people have done some other work on this, and we've talked about some of these sterile insect technique type approaches and other things like that. But one of the things that this World Mosquito Program is doing that really I think is a big deal is that they are making a centerpiece of their program their outreach to their communities.
Yeah,
because they understand that if you just go in and release these and say, "Hey, people, we saved you from dengue. Aren't you happy?" It's not going to go over well.
Nope.
You have to go in ahead of time and say, "Hey, we propose this. Here would be the benefits. Here would be the risks, and we want to get everybody involved and have a big conversation about it before we do anything." And then they involve the schools, and they have as like this kid posing with the Mozzie box, and that's how you get something like this done. It has to be something that the community comes around to and says, yeah, this does sound like a good idea, and the people who are going to be living with it sign on to it.
We need to hire the people who orchestrate that to take care of the anti-vax issue.
They're busy right now.
One does not simply walk into Boston and build a BSL-4.
That's right.
One does not, and yet one did.
Yes,
and yet one
could have been done differently.
It should have been done. Well, they had a rich history of that too, though, with the first genes that were discovered and that sort of thing. So there's sort of a reactionary group that's there as a result.
And also, I thought another cool story arc this year was the evolutionary history of RNA viruses. So this started with TWiV 491 in April, the Ilium and the Odyssey. It's a cool title.
Yes.
And this was a really neat paper where it was a Nature paper where they had, it's called the Evolutionary History of Vertebrate RNA Viruses.
Right.
They had sampled a whole bunch of vertebrates that are not typically sampled for viruses, like reptiles, amphibians, lungfish, ray-finned fish, which I just get a picture of a fish wearing sunglasses, right? Ray-Bans, cartilaginous fish, and jawless fish. They found 214 new viruses and could establish a long evolutionary history. Do it by doing that. And it shows that if you just keep looking, you're going to be able to fill in lots and lots of
gaps. Did they look at the coelacanth by any chance?
No, I don't think there's a coelacanth in here.
Okay, because that's one of the very, 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 very oldest of fish.
I know.
Isn't the coelacanth related to the lungfish? Or is it?
It may not. You might be right, Alan. I think it does have some distant relatives there.
However, what's cool about the coelacanth is that they have found endogenous retroviral elements in its genome.
Yes.
Wow.
And how old are coelacanths?
I mean, they're back in the Devonian era.
And you can still find them.
Yeah. They're alive.
Do you ever catch one, Dickson?
I tried. No, they weren't running when I...
You need a long, long line.
You do.
Do they explode when you pull them up?
No.
And
apparently they taste really bad, though.
But there are 2 of them in the Museum of Natural History that you could sample right now if you wanted some stuff.
Are they alive?
No, they're resting peacefully in a
that's
large lead container of formalin.
That makes them a little hard to sample.
Why is it lead?
I don't know. That's a good question.
Are you leading me on?
I wouldn't dare do that.
And then the other one that links in with that is 526, which is a more recent one. In the beginning, there was Paul, where Eugene Condit and his colleagues took those data and many others and put together a beautiful timescale of evolution of RNA viruses.
Yeah, that's a neat paper.
Which we did recently, and I'm sure is fresh in everyone's mind, and it's just wonderful. Starting with the RNA world and who came first. It's just great. And
I enjoyed that.
And that the data from the original paper that I just talked about was part of this data. They took it and added other sequences, thousands and thousands. They focused on the RNA polymerase, which is in common to all of these sequences. Very cool stuff. And there'll be more of this because people are going to keep looking for viruses and analyzing the sequences. So looking more forward to that in the next year. Rich, you probably picked the next one, right?
I did. I was one of my favorite episodes was actually, I think this is Dickson's title, "A Pox on Your Horse." TWiV 478 that
might have been
highlighted.
No, that's Alan. That's Alan.
But it's Dickson's choice as a favorite title.
You got it.
Okay,
that's what it is.
It highlighted the role of horsepox in the smallpox vaccine, plus a technological achievement that I'll get to. But the smallpox vaccine, its actual natural origin has always been, has long been a mystery, because it was developed, decades and decades before we really understood any virology. And when people finally had a close look at it, it didn't resemble anything that was actually any pox virus that was actually circulating in nature. There's always been a suspicion that maybe it was related to a horsepox that had since gone extinct. And there were a couple of reviews and a couple of papers on this episode that gave credence to that idea. So it looks like maybe a now extinct horsepox virus could have been the original ancestor of the virus that was used in the smallpox vaccine. At about the same time, and as a matter of fact, we saved this episode until this was actually published, Dave Evans' laboratory in University of Edmonton in Alberta published a paper where they, from entirely synthetic DNA and using techniques that we won't go into, managed to synthesize horsepox, that same virus, from scratch. This, by the way, is a virus that has vaccine potential and is not going to kill any humans anytime soon. But the synthesis of that virus from scratch, it being a pox virus, and any kind of this kind of synthesis of something that sounds like a pathogen generates a lot of excitement, and that did indeed generate a lot of excitement. The press got a hold of that and ragged on it for quite a while. At any rate, it was quite an achievement, I think. And since, interestingly, has the fuss over that has kind of died out till the next time.
Yeah, I think it's cool that you can build something so big. And I think in that paper, they were buying oligonucleotides, tens of thousands of bases long.
Yep.
And then throwing them in cells, and out came virus.
Yep.
Amazing.
So a little more complicated, a little trickier than that, but that's essentially correct. Yeah.
So, Rich, are you saying that the press wanted to scratch a horse virus?
I just looked up why it's called from scratch. I didn't know the etymology, and it's that from the scratch drawn in the ground, serving as the starting line of a foot race. A runner starting from scratch received no handicap, starts at the beginning. Because I'm listening to this guy from scratch.
Right.
So when we published the infectious polio cDNA back in 1981, Harold Schmeck wrote an article for the New York Times, and he called it poliovirus synthesized from off-the-shelf chemicals. He didn't say scratch. Off-the-shelf chemicals, but not really. You can't just go on a shelf and get nucleotides, right?
Not really.
Not in my pantry.
So we had Ryan Noyce on our Halifax TWiV. Alan, do you remember Ryan Noyes?
Oh, yeah.
And he talked about this, and gave his own little personal spin on it. So that was cool. That was nice to have both. So we can do a picornavirus, which is about 7.5 kb in length. We can have it synthesized for about $5,000. The whole thing in one bit.
It's amazing.
So if we can't find a virus, and we have trouble finding infectious viruses, we can just, of course I can't do that at will because I don't have a lot of money to throw around, but you can recover viruses if you can't get them from, oh no, we're not sending you this virus. Okay. We'll just make it.
I think the price tag on the sequences for the Horsepox was about $100,000, but that doesn't account for all the time and labor, paying the people to actually put it together.
Yep.
Right.
Plus the funding of the laboratory. That's just the materials.
Now, Kathy, you liked the Ebola story arc.
Well, actually, I just inserted 2 parts. I think Rich had already picked it.
Oh,
yeah, I picked that one. Yeah. Well, I was focused mostly on the fact that Ebola epidemics continue to happen.
Mm-hmm.
Interestingly, we don't make as much. Not just we, the world in general, the press doesn't make nearly as much fuss about it as it used to. But I think it is noteworthy that to this day, there's still an Ebola outbreak going on in East Africa.
Yep.
And that actually, I wasn't aware until I was reviewing this. It hadn't really sunken in that there was an outbreak in May in Western Congo,
right,
that we covered in TWiV 494.
Mm-hmm.
And that ultimately only amounted to about 50 cases and 30 deaths. And then I don't know if it burned out or was controlled out of existence. But then in August, an outbreak arose in the east in Congo that is still ongoing. If you look at the CDC information and graphs, it looks like it could be declining, but I wouldn't call it contained yet. And it has involved at the latest count 585 cases with 356 deaths. Those are confirmed cases.
And the official start of this was May 8th, 2018. That's when the WHO was notified by DRC Ministry of Health of a couple of lab confirmed cases.
And interestingly, in these cases now, of course, we have a vaccine that's being brought into play. And they're using this in a strategy called ring vaccination, where when they identify a small outbreak, a few cases, they'll vaccinate everybody in the vicinity. And there have been distributed in excess of 50,000 vaccinations. There's no data yet that I can find as to how effective that has been, if at all. But I presume that somebody's keeping track of this, and we'll find out how good the vaccination is.
Well,
at any rate,
yeah, this is a really, really unfortunate situation. Ebola virus outbreaks of this size occur only in places where there's chaos. If you look at countries, this is a virus that spills over to humans occasionally. And if you look at countries that have functioning health systems in Africa where this occurs, they'll have an Ebola virus case, and that person goes to the hospital. Everybody knows what to do. They get contained. They survive or they don't. And survival rates are improving as people understand how to manage this. And that's it at one case. Or they'll have a couple of cases, or occasionally a couple of hospital workers get infected. These big outbreaks that we're seeing now are the result of places that have non-functioning governments and civil wars going on, and nobody knows where anything is or what to do, and everybody's shooting at each other, and that's what's going on in DRC. So, it'd be nice to have some data on this vaccine, but several times they've had to pull the health workers out because people were suddenly getting shot and hacked up with machetes, and that's why this virus is spreading. So until we until we address the underlying human problems, I don't think we're going to be able to see the end of these types of outbreaks.
So,
one of the things that interests me is that an outbreak of this size 10 years ago would have been just huge news internationally for a long period of time. But in particular, since the huge outbreak in West Africa some time ago, the press has become, and the population, I suppose, has become somewhat jaded to this. And I'm always reminded that this sort of my background for this is Lassa virus. There are still annually between 1 and 300,000 Lassa infections in Africa, and approximately 5,000 deaths. And you never hear about it.
Never.
Just
shrug
it off.
Why is that?
It's Africa's equivalent to influenza in this country.
But why don't it kills more people than most Ebola outbreaks?
We have 40,000 people that die in this country. Come
I know,
on.
but
we don't hear about that
why
either.
Ebola? Because people are afraid of Ebola more so than Lassa?
I think so.
Yeah, that's true.
But one of my points here is that as time goes on, people are less and less afraid of Ebola. With this, even the current epidemic going on, you don't hear much about it.
Nor do you hear about malaria.
Kathy, you were
I was just going to say the reason I have two things to point out about this is that at least in two of the episodes, we actually talked about a basic science paper. And so in 494, there was a paper that addressed the question, are there the recent mutations that are in the Ebola virus responsible for the high case numbers in the outbreak from 2014 to 2016? And the short answer is no, that we have to look to lots of other factors for the high case numbers, some of which Alan has just alluded to. And then the second one was in TWiV 482, we looked at a paper that was effectively asking, are there asymptomatic Ebola infections? And this was a paper that was summarized by someone that we met just a couple months later in a commentary, and that person is Ian Crozier. And in the primary article, the page numbers for Ian Crozier's commentary are listed wrong. And so it takes some searching to actually find that commentary. And so I think I missed it at the time. But he did a nice job of describing what happened here. And that is that there have been lots of attempts to put together serosurveys to determine whether people have been infected with Ebola, and we just didn't know it because they had asymptomatic infections. But there were a lot of problems with those kinds of things. But this particular paper looked at samples collected over 2 months in 2007. They used a standardized assay, an ELISA assay. They had a very large population size with an appropriate control group. They had 2 randomly selected rural villages. They had non-outbreak controls. And it was at the same time and not too far geographically distant from an existing outbreak. And some of the outcomes of this serosurvey were that 11% of people were seropositive that seemed to have been asymptomatic. They weren't aware of having the disease. And they were able to, for the first time, actually make some correlations. The higher seropositivity correlated with more visits to the forest, having exposure to rodents, but no associations with exposure to non-human primates or to bats, which is interesting. And there was an association with males and older individuals. And so then Ian Crozier does a nice job of giving some possible explanations for the heterogeneity of results that are in the literature. Maybe there's an unknown antibody that cross-reacts with Ebola, or maybe their antibody that's being detected is to an undiscovered mildly pathogenic or non-pathogenic filovirus, or the one that's kind of how the paper comes out in that is that asymptomatic Ebola virus is a common thing, about 11% of people having it. But then he says the caveat is that classifying an infection as asymptomatic is really difficult for the patient, for the clinician, and the researcher. And also there's the possibility that maybe people were exposed to just Ebola virus antigen and not to viable virus. And they could develop antibodies that way. But I thought it was a nice way of trying to address a longstanding question about, are there infections that we're just not seeing, maybe because people have genetic resistance or something like that. And at least we now have this baseline level of understanding maybe 11% or so.
I mean, look at what's happening with Lhasa. I mean, if there are 300,000 cases, only 5,000 deaths. Well, I mean, that's not only, but that's a hell of a lot of people that got infected that didn't die from that. So, among those people that got infected, I'm sure there was a spectrum of illness that ranged from almost not sick at all to almost dead, but then came back.
Almost dead.
So, yeah, well,
it's a Princess Bride reference.
Yes. Exactly.
Not all dead.
It's not completely
all
dead.
I mean, it's not completely dead. It's just a little bit pregnant.
I just rewatched that movie.
I just watched it for the first time. I got it for Christmas.
It's a great movie. Great movie.
We did quite a few episodes on Ebola this year.
Yeah, we did.
Yeah.
So I think it's in line. I think we should have Crozier on TWiV next year.
Oh, that'd be great.
Next year or this year?
That's right. This year. Yeah. I forgot. We're already here because
for the uninitiated, he was an Ebola survivor,
and he was at the ASM TWiV, and we asked him to come on. He's got amazing blue eyes. I remember.
Yep.
Yeah,
that was part of the story. One of his eyes turned a different color.
Yeah.
Yes, we'll have to get him.
Along the same lines similar lines that Kathy was talking about in TWiV 505, where we talked about Ebola as well. They talked about an outbreak that apparently started from a persistent infection.
Right.
Okay. So somebody carrying Ebola around for a period of time asymptomatically, and then that sets off a new outbreak.
One of the other themes here last year with Ebola was the significance of these genome mutations that were identified and improve virus entry into cells and culture, right? What role they play. They seem to have been fixed in the West African outbreak. I will be curious to see as we get sequence from these outbreaks, if we do, whether similar mutations are fixed as well. I think that'll be interesting. Although, as Alan says, maybe it'd be hard to get anything out of the country.
Yeah, you can. I mean, I'm sure you can get some data. The question is, can you collect it systematically in the way you'd like? And that's going to be the tricky part.
So when we interviewed David Quammen on TWiV, he asked, "Have you ever been to DRC?" I said, "No." He said, "Very difficult country," because he's been there a few times. And he said he understands why the immunization has been a problem here in this outbreak. And Kathy, I think you had a bunch of additional stories.
I can treat these kind of like snippets, but to me, the things I always find fun are when we get some additional clues to mysteries or longstanding questions. And so one of them is, why are some influenza strains more virulent? And in TWiV 481, this was looked at, and this was the paper where they looked at defective viral genomes, also known to those of us who learned virology longer ago as defective interfering particles. And what they found was that the presence of more particles with these defective viral genomes correlated with decreased pathogenicity in mice. And so this was something that was kind of appreciated in cell culture, but this actually looked at it specifically in mice for influenza. And so there's this inverse correlation. If you have more of these defective particles, then you have less pathogenesis. So I thought that was cool. We've always talked about, how bats carry so many viruses, and there's some variety of reasons that have been postulated for that. They have a high metabolism because they fly and all this other stuff. But there have been papers that have indicated that bats have changes in innate sensors for DNA, including TLR9, IFI16, and AIM2. And now this paper that was in TWiV 484 identified that STING, which is an innate sensor for DNA, has a single amino acid change in a highly conserved position relative to other mammalian, bird, and fish stings. And that single mutation leads to a reduced interferon response. And they looked at 31 bats, and they all have a different amino acid at that position that's not, in this case, a serine. And then the really cool thing is that they were able to mutate the sting to have the serine, and that caused increased interferon production and reduced virus replication. So it was just a nice answer, possible answer contributing to the aspect of, bats and all the viruses they have.
That's very cool. I love that. Yeah.
Yeah. And then a paper that was addressed a question in norovirus. This is from Stephanie Karst's lab. Why are there really high levels of norovirus shed in the feces of infections, but only low numbers of infected cells have been found in the GI tract? And so they postulated that perhaps this is because there's pockets or hotspots of virus replication. And they used the mouse model, which is in this case an immunocompetent mouse model for mouse norovirus. And they used highly sensitive technique. So it was, to me, the technology that really drove the answer to this mystery, or part of the answer to this mystery. So this technique called RNA scope, which is based on hybridization. So it's really sensitive. And what they were able to show is that immune cells in gut-associated lymphoid tissue are really highly infected. And so that included macrophages, dendritic cells, and B cells, and also T cells for the first time. So that was kind of a new finding. And the nice thing is that these data are consistent with biopsies of immunocompromised people in which most noroviral antigen was found in the intestinal lamina propria, in other words, immune areas where there's a variety of immune cells. So it was kind of a nice answer to that question. It was, as I said, driven by the technology. Then, of course, as a DNA virologist, I really liked the paper from Dan DeMeo's lab, where they showed, this is in TWiV 515, that human papillomavirus L2 protein, which is a virion capsid protein, the virus being non-enveloped ends up inside the endosome, but it has to get out of the endosome to traverse through the rest of the infection process. And this L2 has a sequence at the end of it that I just forgot the name of the something peptide. Help me out, guys.
I'm looking.
Yeah, I'm looking too.
I never forgot it because I never knew it.
It's like it's the poking through the
cell penetrating peptide.
CPP.
They have the cell penetrating peptide, so that pokes out from the endosome and engages with the retromer, which is in the cytoplasm.
Very cool.
And that then allows the virus to get transported through the trans-Golgi network. So check that story. Yeah.
You were on that episode, Dickson.
Yeah, well, I didn't claim that I wasn't on it. I just forgot it.
Oh, I thought you said you weren't there.
I didn't say that. Never said that. No.
Okay.
Go back and check the tape.
My apologies.
Right. And then a mystery that we maybe didn't even know existed, unless you thought about why are there so many AUGs in the, upstream of the open reading frame in poliovirus, is what is the function of a conserved upstream open reading frame in the enteroviruses? And the answer is in the title of the paper, making it a snippet at this point. An upstream protein coding region in enteroviruses modulates virus infection in gut epithelial cells.
That's such a cool paper.
Yeah, really fun to find.
So that would be an unknown, right?
Right.
Yeah, and just I know we said this on that episode, but it was just so cool that this group of viruses that has been so intensively studied. I mean, polio, there's like all this research from the early days of virology onward, and then the broad universe of the enteroviruses, and everybody's been staring at these genomes for years, and there's this whole gene. That's there.
Yeah.
And if you haven't looked at the image for that episode, check it out.
Yes. That's a great show image.
Because there's a famous person actually looking at the open reading frame.
Oh, right.
Frame, yes.
Dickson, you weren't there.
No.
Rich, I think you did some of the vaccine stuff, right?
Yeah, I put this in, or most of it. There's been some
put some
additions. But so we're always talking about vaccination on TWiV, but there were, I think, 3 episodes that had a particular focus on vaccination. One was our TWiV 500 episode where you had invited Ginny Sue from the Austin area, who's the, I guess, founder and chief, the point person in an organization called Immunize Texas that promotes vaccination and tries to fight off the anti-vax crowd. And she, I thought, delivered a wonderful summary of the whole vaccination, anti-vaccination issue in that episode. So I think that is as nicely a coherent synopsis of the whole anti-vax thing as you can find in that episode, TWiV 500. Then we had, when Vincent and I went out to Irvine for a conference that was organized by Bert Semler that had Stacy Schultz-Cherry as a speaker and 2 other individuals whose names are escaping me right now. We can look them up, about vaccination. And that was all, about the issues with vaccination as well. That was very good. Oh, the other guests were Douglas Diekma and Andrew Neumer. So that's another good resource for discovering that. And then most recently, the special that Vincent did interviewing Peter Hotez on the occasion of the publication of his new book, Vaccines Did Not Cause Rachel's Autism. And I thought he had a particularly poignant perspective on this.
Yes.
Because, of course, one of the myths or lies circulating is that vaccines cause autism. And Peter, in addition to being a scientist, is the father of an autistic child. And so he can look at this from all angles. And his book apparently explores vaccinology and the anti-vaccine efforts and also the actual causes, what's known about autism, and his experience as a parent of an autistic child. So all 3 of those episodes, 500, 496, and the special episode with Peter Hotez, in addition to other conversations we've had, are a good focus on vaccination.
Yeah. And the Peter Hotez episode, by the way, is a 19-minute TWiV.
Yeah, that's an easy one.
There's
a world record short. It's a
no weather.
Very,
I don't think any weather.
Very, thoughtful and good episode.
This Ginny Sue, I could actually take that out as a standalone,
Yeah.
Mm-hmm.
Right.
It would be worth publishing because she said some really good things. And also, the other 2 are great also. The Irvine one, they said lots of interesting things as well. So I'm interested in this measles outbreak in the Ukraine, which has been ongoing this year. And there's a nice Lancet article called Measles War and the Healthcare Reforms in Ukraine. And it starts, Russian-backed attempts to sabotage Ukraine's economic, political, and health developments have left the country fighting a measles outbreak and continuing a bloody undeclared war. And they've had over 23,000 cases so far in Ukraine, with many deaths. And the Kremlin is supporting social media accounts spreading discredited theories about the MMR vaccine. So that, combined with shortages and underfunding, have been blamed for the outbreak. Can you believe this? The Kremlin is pushing forward these nonsense stories about MMR and autism just to screw Ukraine over. And of course, it's spilled over from Ukraine into much of Europe, and there's been a lot of measles in Europe. In fact, if you go to airports here in the U.S., you'll see signs, make sure you're vaccinated because there's measles in Europe. It's unbelievable. Come on, Putin.
Yeah.
Good luck with that.
Yeah.
Do you think they've had any input in the United States?
What do you mean?
The Kremlin? Oh, yeah.
Oh, yeah.
Oh, yeah.
On this subject.
Oh, yes. Actually, I tweeted a link about that a little while ago that one of the things in the run up to the 2016 election, they were the Russian propaganda machine was not just sowing discord with the political stuff. They actually were pretty much finding any kind of difference that they could use as a wedge, and a lot of those accounts were promoting anti-vax stuff too.
I presume climate change will throw it in.
Oh, sure. Oh, yeah, absolutely.
Sowing discord.
Sowing discord.
That was the agenda.
What you sow, so shall ye reap.
What's amazing is that this has become a political issue, which is just crazy.
It's abhorrent.
But people are there's an anti-vax group in Texas. I forget what their name is, and I don't really want to remember.
No, don't.
But if you look at their website, it has nothing about what vaccines do, or any health information, or even anything about safety. It's all about your right to choose.
Right.
And this, and promoting, one of the common memes is "Don't mess with Texas." Texas has its own sort of Texas Republic independence thing going on.
They do.
And this sort of hits on that, saying nobody can tell us what to do, right?
That's right.
And some political groups have engaged with this to try and get support for particular candidates that it really has nothing to do with healthcare or vaccination. And it's so annoying because it does such damage.
It's true.
And kids get the brunt of it because they get sick. They get measles, right?
And they don't have a voice in it.
No.
No, you notice the states that have no laws about wearing helmets in motorcycles.
Oh, yeah.
Those are the same. That's the same group.
Mm. Kathy.
Well, I just wanted to point out that Vincent put in his blog post links to the Spanish and English versions of a terrific book entitled Paul Has Measles that has really excellent illustrations and text to explain to children about measles, viruses, vaccines, and so forth. And I highly recommend it to everybody. Just check it out.
And there are other translations in progress, and we'll let you know about those.
Oh, cool.
I know Arabic is one of them. Islam Hussein is involved in that, but there are others. There are many others, so it's pretty cool. We'll
Hopefully Russian is going to be on the list.
I would hope so. Another arc I thought was cool had kind of revolved around the Outbreak episode that we recorded at the Smithsonian. Right. Smithsonian has this awesome exhibit, Outbreak, Epidemics in a Connected World. It's going to be there for a couple of years. It's in Washington, D.C. It's free. Go see it. It's awesome. I'd go again and again because to see viruses being the focus of an exhibit is pretty unusual, and many millions of people are going to see it, and they did a really good job. So that episode 501, Outbreak, is really cool in the museum. But then we started doing every now and then, snippets on some of these outbreak viruses. And the most recent, we talked about hantaviruses in TWiV 527, city mouse, country mouse, influenza virus on TWiV 520, Ebola viruses on TWiV 507, and Nipah virus on TWiV 504. So I think we usually spend about 20 minutes. Well, maybe not. Depends. Talking about
some time. Yeah.
Each one and why it's a zoonotic virus and so forth. And it's tied in with the exhibit. And so these are all viruses that were part of the exhibit.
There's still plenty to go, Vincent.
We have.
Are they going to be relapsing?
I'll tell you which ones we have left to do. I keep a track of that. MERS coronavirus, SARS coronavirus, HIV, Zika, and smallpox. And we got a lot.
Okay. Wow. Great.
So we got to have, you'll have to be here for that smallpox one.
I'm here.
Professor Condit.
I'll do that.
We'll get an HIV person to do an HIV one with us too. Right. There's fun. They're a lot of fun. I like that a lot.
Yeah.
What's the matter?
Steve.
Goff?
Yeah.
He's going to be a guest actually.
Or Dan.
Next Friday.
Dan Griffin. Yep.
Steve Goff.
Nice.
He's going to do their latest paper, so maybe we could start with a HIV snippet, or maybe it's too soon. Another arc which I thought was cool, insect viruses and insect immunity.
This was one of my favorites,
too.
Yeah,
me too.
The most recent ones TWiV 525, The ABCs of Bee Viruses with Michelle Flanagan.
Lots about bees.
Yes.
A lot
Great
about bees.
stuff.
Honeybees. You can learn a lot about honeybees and their behavior.
Really interesting guest, and I like that show a lot.
And then how they infect them with viruses, what they do. And I got an email from her husband, whose name is escaping me, but he is also a scientist out there in Bozeman, and he offered to take me into the back end of Yellowstone to see all the cool places where they sample next summer or next fall. And Brad, I think is his name. Yeah, of course, everyone can go.
Love to go.
He golfs with David Quammen.
Oh my!
What a small world, right?
Well, what a small town, anyway.
Not so small anymore. I used to go fishing out there. I started in 1982. Bozeman was our destination, and we spread out from there. And I have watched that place grow. It tripled in size since 1982.
Oh, wow.
It's a big retirement community for military, and it's now a big retirement community also for academics. But it's still a very pleasant place to go to.
Trying to find his name. I don't want to have his wrong name here. This is not good.
There's plenty of time to correct it.
Blake. His name is Blake.
And what's his specialty?
I don't know. Golfing with David Quammen.
Golfing with David Quammen.
And he was a postdoc with Jennifer Doudna, and he knows Sam Sternberg, who's here, and I did a TWiV on CRISPRs with him. It's all connected. Anyway, back to the topic. We don't usually go off like this. I'm sorry.
Yeah, no, we don't.
You didn't go up.
Right.
We never digress.
Slicing and dicing with Raul, TWiV 522. He was here visiting, and we talked about cricket paralysis virus, a protein. Now, cricket paralysis virus is a picornavirus. I wonder if it has that extra open reading frame. Probably not. It's not an enterovirus. Anyway, it encodes a protein called 1A that restricts RNA immunity in insects. It interferes with Argonaut protein, which is part of the RNAi machinery. It not only binds, one part of the protein binds to Argonaut and blocks the active site, and the other part directs it to degradative pathways. It's like, if you're not doing it one way, you do it two ways to make sure. So that was pretty cool. TWiV 495, we talked about, this is one of our favorite arcs, how a parasitoid is revealed to hyperparasitoids.
Yes. These are always fun.
These are, of course, polydnaviruses, symbiotic polydnaviruses. So the story here is that the wasp injects these polydnaviruses into a caterpillar host, and these cause changes in the host which reveal that to a hyperparasitoid who will then superinfect that caterpillar, that infected caterpillar, because of cues it gets from the viruses from what they're doing.
To parasitize the wasp that parasitizes the caterpillar.
Isn't that amazing?
Right.
It's just amazing. I love these. Every year we do a few of these parasitoid stories. Another one, 522, Readers of the Lost Orph. We also talked about the first DNA virus from fruit flies, actual infectious virus gotten there. That was very cool. TWiV 485, Fishing with Defective Flies.
I've done that.
You have?
Many times. I once went fishing with a friend of mine, and for half a day, I rose fish constantly and couldn't hook a single one until I looked at my fly, and it turned out that the point had been broken off on a rock.
You rose them, meaning they came to the surface?
They came to the fly, and I struck,
looked,
and there's nothing there. And you said, what the hell am I doing wrong? I'm too early, too late? Nope.
No
Perfect.
hook.
So you say the fish are rising, is that right?
That's right. That's what you'd say.
Like we say Mars is rising.
Yeah, no, that's
right. It goes in the sky.
So the insects that they're after are floating on the surface of the river. So they rise to the
I have
fish.
to say, I learned a lot about fishing. What was it, last summer we went fishing?
Yeah,
no, the summer before.
It was
wasn't
before
it?
that, yeah.
That was a lot of fun. I have to show you those videos sometime.
Sometime.
So this Fishing with Defective Flies, paper from Carla Sally's lab. So cool that, RNA viruses are targets of the RNA interference system when they infect insects. But it turns out that DNA copies of these genomes are made so that you can amplify the RNA that gets involved in this RNA-based immunity. So it amplifies the small interfering RNAs, basically. It's so cool. And it's not just DNA copies, but they copy defective viral genomes preferentially, which, is interesting because, as Kathy talked about earlier, defective genomes seem to modulate the virulence of flu. So there's some theme here going through all of this. That's very cool. Another one, 482, don't evome without antibody-expressed endogenous viral elements in the mosquito genome. They sequence the mosquito genome, and they find sequences of viruses. This is from Raul Andino's lab. And they're wondering if these are serving. So these are RNA viruses that infected them. They were reverse transcribed by endogenous reverse transcriptase, integrated into the viral genome. And they serve as defense. You can make small interfering RNAs from them and prevent virus infection. Insects are
almost like CRISPR for flies.
I was just, or restriction enzymes for bacteria.
Yeah, similar ideas, not the same systems, but there are differences, but similar ideas. Exactly.
Amazing.
Right. Analogous, not homologous.
Analogous, not
there
homologous.
There you
Good
go.
title for a show one day.
Exactly.
And then the last one, which was way back in February, Trypsin the flight fantastic. This is very cool. A fungus in the gut of mosquitoes that helps dengue virus to replicate. How cool is that?
So it's an anti-Wolbachia fungus.
No, it has nothing to do with Wolbachia.
Right. Unrelated.
It blocks trypsins that would normally degrade dengue virus.
Nice.
What a wonderful interconnected world.
Amazing.
Right?
Amazing.
And finally, the last kind of arc is really two things. First, huge RNA virus genomes in TWiV 509, planaria and aplysia. Huge, over 40,000 bases. Now, these are sequences. They don't have virus.
And weren't these coronaviruses or
coronavirus? Yeah, they were nidoviruses. Yep.
Okay.
They were big and discovered by just searching through sequences and databases.
Right.
So giant RNA genomes and, of course, giant viruses. A year cannot go by. We don't talk multiple times giant viruses. TWiV 490, shave ice and giant viruses. This is a new Mimi virus from green algae with genes encoding the enzymes of fermentation. TWiV 506, a cafeteria full of jelly rolls. This is a giant virus that infects Cafeteria rowenbergensis and has a cool way of assembling. TWiV 521, spitting in the alley. Another giant virus from 30,000-year-old Siberian permafrost. Mollivirus sibericum, and what we learned from that. And one more, TWiV 484, float like a Mimi virus, sting like a bat. Tupan virus, with the longest tail in the known virus sphere. It's a really cool looking virus. It's all fuzzy and has a spherical capsid and a tail coming out from it.
Yeah, it's almost like a one of those square bottom ice cream cones.
Yeah, with a nice little ice cream ball on the top.
Yeah.
So giant and they're just so interesting. It has the most complete translational apparatus of the known virus field, and as the authors say, only the ribosome is lacking. In their wonderful French style, right? Only the ribosome is lacking. Seulement le ribosome manque. Something like that. And that's it. That's our cool stories of last year.
Yes.
And by the way, folks, Ray Ortega responded and sent a link to the TWiV survey. We've got 212 people so far. Pretty good, but we want 1,000. So when you get home,
if you haven't taken the survey,
please take the survey. asm.org/TWiV poll. Most people who listen to TWiV are not members of ASM. Very interesting.
Yeah.
Should ask if they're ASV members, right? So most people subscribe. Then we have what topics. Most people listen to every episode. Very cool.
Yeah.
List
of topics doesn't include weather. But 15% like other, so I'm going to think that's maybe the weather.
Maybe the weather.
Most people are satisfied with the variety of topics. Oh, look at this. 80% feel that the length of each episode is about right.
Yes.
Oh. Only 15% feel it's too long. Dickson, how likable do you find the hosts?
I love them all.
75% find us very likable. And production quality is great. Yeah, so thank you. Keep going. We want more. There are actually 1 or 2 replies of people who do not like the hosts. One person strongly dislikes the hosts.
All of them?
Just says host. 1 dislikes and 6 neither likes nor dislikes. Makes me so sad if you don't like or dislike me. Pick one.
Pick one. That's
all right. I ask everyone to give us their favorite titles of 2018.
Right.
What do you think, Dickson? What's your favorite title?
I like the Pox on Your House.
Pox on Your Horse?
Yeah, Pox on Your Horse. I loved it.
Rich?
Did
you leave? Oh man, you missed so
Sorry,
much.
I'm here. I was
He's just listening. He's just muted, probably.
I'm recovering from a cold, so you don't want to hear that.
That's true. That's true.
Readers of the Lost Orph, TWiV 522.
So mine was Raiders of the Lost Ark, TWiV 477. Alan?
I liked Biles to go before I delete.
Kathy?
I chose 419, the Ilium and the Odyssey, but I had several close seconds. TWiV 502, Texas Road Phage, because that's when we went to Texas A&M and learned all about phage, and it was a road trip. And I also liked A Cafeteria Full of Jelly Rolls, TWiV 506.
Yeah. I have to say, this Raiders thing, also on TWiV, we had Raiders of the Lost Orco. That's about raider ants, and they've made a mutation in the orco gene, which controls olfaction.
I hope we don't get a letter from a movie studio.
And,
or a killer whale.
And Raiders of the Lost Ark, that was an Ark gene.
Yep.
I think it was supposedly donated by a retrovirus. And then, of course, Readers of the Lost Open Reading Frame. And speaking of olfaction, I already gave you the answer. Crap. I got this magazine in the mail today about Columbia Nobels.
Yep.
It's called Test Your Metal.
Yep.
Ha ha, you get it?
Yes.
Duh.
In 2009, Barack Obama, who graduated in 1983, became the 4th U.S. president to win the Nobel Peace Prize. The first was Theodore Roosevelt in 1906. What did he win for? A, opening up the Panama Canal. B, negotiating an end to the Russo-Japanese War. C, establishing the U.S. Forest Service.
C.
Is that right?
I don't know. I'm guessing.
Let's see. The answer is no. It's negotiating an end to the Russo-Japanese War. Here's another one. In 2004, 2 Columbia scientists, Richard Axel, 67, Columbia College, and Linda Buck shared the Nobel in physiology or medicine for identifying a family of genes that govern what? A, olfaction, B, vestibular system, C, the auditory system.
A.
That's right.
This is cool. Is there anything else I can ask you here? I actually don't know the answers to most. Okay, in addition to Axel and Lee, 8 current Columbia faculty have won a Nobel Prize in 6 fields: peace, physics, physiology or medicine, chemistry, economics, and literature. Horst Stormer, do you know which one he won?
I would guess economics, but I'm not sure.
Number 10. I can't find the answer. Where is it? Where did it go? But how about Eric Kandel?
Well, physiology and medicine.
Joseph Stiglitz? I have no idea. Joachim Frank?
He was the one for...
Stiglitz was physics, wasn't he?
Don't know.
Chemistry, isn't the last one chemistry? The cryo-EM.
Oh, okay. So Stormer is physics.
Stiglitz is economics.
You're right. You're right.
Economics. How about Joachim Frank?
He's the cryo-EM person.
Yeah,
right.
Chemistry. Martin Chalfie?
Biology, medicine, and physiology.
Chemistry, actually.
Chemistry for green fluorescent protein.
Orhan Pamuk? No idea.
No idea.
That was literature.
Literature.
All right, one more. George Wald, 28.
It's vision.
Graduate, won the 1967 prize Physiology or Medicine for his discovery that the retina contained traces of which vitamin? A, B2, or C?
A.
Hey.
Yeah. Okay, you guys are all.
And then he went off on a toot.
What does that mean?
That means he started to pontificate about crap that he didn't know anything about.
No.
We all do that.
No, well, we're going to get to that.
Well, I don't want to
a pick of
implicate.
the week.
I'll implicate myself and you.
No, Marty Chalfie once was asked, what's the difference between your life before and after winning the Nobel Prize? He says, now afterwards, everybody believes everything I say, which he says they shouldn't.
All right, I want to read a letter from Witama, our friend in at James Cook University. She's, I believe, in Australia, right?
Mm-hmm.
It's a PhD candidate there. Hi, TWiV team. I thought 2018 was an excellent year for TWiV. I've attached a short compilation of some of my favorite clips. I'm looking forward to 2019 when we get to hear more science, more Brienne, and more weather. Now, let me play this.
Oh.
See if I can get this technology to work here.
Oh, dear.
You haven't heard this, Dickson,
I
right?
have not heard this.
Everyone else has. Are you ready? Welcome to TWiV, this
Thank
week
you.
in virology.
The kind that make you sick.
Boy, do we have weather.
Mild winter day.
Wispy clouds.
Kind of hazy.
It's a little chilly.
I'm looking at the window and it's cloudy.
Pretty brisk.
23 inches of snow.
Cloudy.
Mostly cloudy with little blue tinges to it.
Mostly sunny.
Puffy white clouds.
28 Celsius.
Chance of heavy rain showers in the afternoon.
Intense heat of summer.
It's very gray and cloudy.
Foggy, rainy.
Kind of cool, maybe thinking about becoming autumn one of these days sort of weather.
A couple of clouds, but otherwise bright blue sky. It's gorgeous.
It's great weather here. 32. So you guys can do the conversion.
Vincent podcasts like a pro, though his age is not something you'd know, 'cause his voice is as crisp as a young will-o'-wisp, but his telomere's running on low.
Put the mic in front of your face.
I will eat DNA regardless of sequence.
Why do we have to pay to publish? So why is this interesting? Let's talk about a paper.
This is science. We're doing science.
I can't afford to publish.
This happens. Fine.
Just a little pedanticism. It's ridiculous.
Give me a break.
So maybe there's a gene for that. We don't have a database of dinosaur sequences.
And that brings up another question I had.
I'm going to either die or change my view. What the hell is that?
How does that work?
What do we want? Peer review. When do we want it? Now. Shame on you, science. Dihydroxy chicken wire. More of a guideline.
Are they virologists?
This is fascinating.
Take him to the opera.
It's an education.
Back to the nomenclature.
It's a single open reading frame.
Open reading frame.
Ebola.
I think it's a brilliant illustration of how you do some fundamental virology.
An absolutely amazing experience.
The TWiV bump.
By far the most boring amino acid out there.
Theta are plural.
Kind of like a caw, like a crow. Caw.
These little teeny flies.
If you do this with ferrets, somebody comes and shuts your lab down.
Yes, sure thing. Always a good time. That's my line.
Thanks. This was a lot of fun.
That's hysterical.
I presume you guys heard that, right?
Oh, yeah.
No, I didn't hear anything. But I had listened to it before.
You didn't hear anything?
No, I just sat silently through it because I knew it was playing through Dickson's headset because I hear him laughing.
It's good stuff.
But I had listened to it before, and it is awesome. Watama, excellent job.
Really well done.
Clip.
And as Vincent says, fully 30% of it is weather.
Is weather, yes. The first third of it is weather.
It's funny. This reminds me of the Smothers Brothers show, which they used to take in 30 seconds with pictures that flashed so fast that you could barely see them. They captured an entire subject from start to finish, like the invention of the automobile or the discovery of Mars or something. I'm just making this up, and it would be played to music. Mm-hmm. And the pictures would flash as the drums were rolling and stuff. And hearing us just like this is compressing an entire year's worth of euphemisms and
yes.
gee whizzes and stuff. I love that. I mean, it really does characterize the group. I mean,
we should have this on the Microbe TV page.
No question about it. I want to hear this again at my leisure.
Yeah.
I want to actually play this for Marlene and say, this is what I do when I do TWiV. When I do TWiV, this is what happens.
I mean, I've always thought about doing something like that. I just don't have time to go through. You have to go through episodes.
Yeah, you do.
I like this part, right?
That's right.
This is wonderful. Thank you, Watama.
Yeah, absolutely.
Let's do some email. Rich, could you take that first one?
Sure. John writes, "Hi, TWiV team. Happy New Year to you all. May there be many more to come. I'm holding out for the 1,000th episode of TWiV. Thank you for an evidence-based podcast that places critical thinking and curiosity at its core. I've just listened to Vincent's interview with Peter Hotez, and it really highlighted to me how important it is to make communication as relatable as possible to your target audience. I'm currently working as a scientific representative and have had to learn my communication in a seat-of-the-pants manner, to quote Peter. In my day-to-day working life, I have to explain scientific concepts such as scanning electron microscopy to customers from a range of socioeconomic and educational backgrounds. Based on this experience, I've found that analogies can be quite useful in getting ideas across. Even customers with many years of education often prefer this as it takes them away from the stresses of their daily lives and gives them something simple and relatable to conceptualize. As an example for explaining the risks of contracting measles versus the risk of vaccine-derived adverse events, quote, there are adverse events associated with vaccination. However, autism is not one of them, and the risk of an adverse event is very small compared to the risk and consequences of contracting measles. You buckle the seatbelts of your kids, even though you know that there is always a risk of a car crash. Not vaccinating your kids increases the chance of serious health problems in the same way as not buckling your kids. End quote. Good example. This isn't a perfect example or a new approach, only something I've learned along the way. I understand that concerns regarding adverse effects would increase once the risk of contracting an infectious disease falls below said adverse events. In that case, it's very important to communicate to the world's citizens that adverse events require better vaccines and support for research into these, not canning vaccines entirely. You don't throw away your car because one of the brakes fails. You get a better brake, and the car is safer as a result. Another good analogy. Communication is itself a tool, and what matters is how it's used. A hammer can be used to commit a murder, or it can be used to help build a house. Finally, you have all helped me start a part-time master in bioinformatics. Thank you, and keep up the great episodes. John from Melbourne. P.S. I like the weather at the start of the episodes. All climate haters can skip it thanks to Jolene's timestamps.
All right.
Good. Thank you. Nice letter. Nice analogies.
Good luck with that master's.
Yes.
Yeah, right.
Kathy, can you take the next one?
Sure. Jonathan writes, "Hey, Twivers. I'm a relatively new listener, but I wanted to follow up on your discussion about prions and corneal transplant from episode 526. First, a little about me. I'm a DVM student at Tufts University's veterinary school. I actually just started working in Dr. Runstedler's lab, where I intend to pursue my PhD. However, prior to vet school, I was a director and laboratory manager at an eye bank. There are a lot of reasons someone could need a corneal transplant, but some of the more common ones are infection, trauma, post-cataract edema, Fuchs endothelial dystrophy, and failed corneal grafts. In the U.S., eye banks are generally regulated by the Eye Bank Association of America, the FDA, and potentially by their state. Screening eye donors is an intensive process, and several guidelines were put into place in order to eliminate high-risk CJD, VCJD patients. I can't remember what the V stands for.
Variant.
Variant. Okay, yeah. The screening process involves reviewing medical records, a physical exam, serology testing, and a donor risk assessment interview, which is completed by someone who knew the decedent well. Obviously, anyone with CJD cannot donate for transplant. However, the regulations go far beyond that. They exclude anyone with dementia, anyone with a degenerative neurological disease, or delirium of unknown etiology. They exclude people who have received a non-synthetic dura mater transplant, human pituitary-derived growth hormone, and anyone with blood relatives diagnosed with CJD. Perhaps the most frustrating regulation excludes people with specific travel history. For example, anyone who spent 3+ months in the UK between 1980 and 1996, or anyone who received a blood transfusion in the UK or France between 1980 and the present. The list goes on and on. Considering that some medical records can be 1,000+ pages and people travel a lot, screening can take a considerable amount of time. Obviously, there are several flaws to the screening process. As you pointed out, this still wouldn't eliminate people in the early stages who die of trauma, so it is exciting to have a potential test. That being said, it is a bit misleading to tell people that they will now be able to know if their corneal transplant contains CJD or not. While this paper shows testing can be done, that is different than the technology being available for widespread use. There is still a lot that will need to be worked out, such as what labs offer testing, how the test will be standardized and certified, the price of the test, how the regulatory organizations will handle testing, etc. The current reality is that patients won't know for sure if the cornea is free from CJD. However, given the current screening regulations and the low incidence rate, the risk of acquiring CJD from a corneal transplant is very, very low. It will be interesting to follow how everything plays out. Thanks for such an interesting podcast. Happy New Year. P.S. I've attached a few documents for reference. One is a suggested donor risk assessment interview. These are the questions someone close to the decedent would have to answer. I've also attached a copy of the EBAA medical standards, which clarify many of the contraindications for transplant if you are further interested.
And we met Jonathan when we were there, Vincent?
I don't think so, no, but we should have, right?
Yeah.
Yeah. Just the things that when he started with the dura mater transplant, human pituitary-derived growth hormone, all those kinds of things, and the amount of time spent in the UK and so forth are what you have to answer if you're donating blood. So just all rang very familiar to me.
Someone I know, I don't remember who it is, said they can't donate blood because they lived in England during this time. Mm-hmm. That's it. You're done. Mm-hmm. I hope we didn't say that the test is available today. I don't think we did, but we know that it requires a lot of development to go from the paper. But now it is potentially developable, and I think it will be, because even though you have all these questionnaires and guidelines, there's still going to be some you miss, and you don't want to miss a single one. And I think the paper showed that if it's in the retina, it's in other parts of the eyes as well. So that's the whole point, because obviously you can't test the cornea because you'd have to break it, right? You'd have to take a piece of it. So I think it's going to be a while, but it's there.
But
anyway, thanks,
is good
Jonathan.
to know.
It was really
thank you,
letter.
Jonathan. Yeah. This is great background on that, and it's good to know that the regulators are keeping a close eye on it.
Ooh.
All right, one more, Alan. Could you take the last
Sure.
one?
Ruan writes, Dear TWiV, greetings and Happy New Year from Cape Town, where it's pretty windy and 4 degrees Celsius, below the ideal temperature for growing enterovirus D68 in cell culture. The recent discussion of hemorrhagic fever-associated viruses and the trend of naming them after places reminded me of the interesting case of Lujo virus, the new kid on the old world arenavirus block. It is interestingly named after 2 places, Lusaka, thus Lu, in Zambia, where the index case originated, and Johannesburg, thus Joe, making Lujo virus, in South Africa, where the index case was airlifted to. Disease caused by this virus during the 2008 outbreak was characterized by nosocomial transmission, hospital transmission, and an exceedingly high mortality rate of 80%. Wow.
I didn't know the name was that. What would you say?
Hybrid.
No,
a portmanteau?
Portmanteau. A hybrid of 2. It's not quite a portmanteau.
I don't think so. Portmanteau is one word is inside the other, isn't it?
Let's look it up. We have to be pedantic.
Word blending the sounds and combining meanings of 2 others. For example, motel from motor and hotel. So it doesn't have to be inside. Yeah. A portmanteau is also a trunk or suitcase that you would put things into.
Yeah, a linguistic blend. Smog. Yeah. There's a famous one. Is podcast a portmanteau? Yeah.
Yeah, sure.
Pod is an iPod, right?
iPod,
cast
right?
Is a broadcast.
And cast is a broadcast.
Yeah. Sure.
In fact, I think I say that in my talk. Or I wrote it somewhere that podcast is a portmanteau. Yeah. I like that word. It's better than hybrid, Dickson.
Okay.
All right. Time for some picks of the week. Mm-hmm. Alan, what do you have for us?
Well, I recently died of flu at Fort Dix.
I'm so sorry.
Yes. So this is a little PR thing that the CDC came out with, and it is quite good. for anybody who grew up in the 80s and maybe played a computer game called Oregon Trail. It was very commonly used in schools to teach about the risks of the westward migration. And there's a very famous game over screen that says, "You have died of dysentery," because that was a very common outcome. Anyway, this one has the 1918 Pandemic Trail game. So it's not playable on mobile devices. You have to play it from a desktop, but it worked fine in my browser. You click on the screen and you play through, and it gives you several. You get a series of choices that you can make. You can choose which role you want to play. I chose to play a soldier, but there's also, you could be a letter carrier, or you could be a farmer, and then there are things that happen in your day. The goal of the game is to survive one day in 1918 at the height of the flu pandemic. So you make choices throughout your day, and depending on how those choices go, you may survive the day or you may die of the flu.
That's pretty cool. I couldn't get it to work on my browser.
Oh, okay.
But which browser did you use?
I'm on Firefox.
Yeah. It didn't work on Safari.
Okay.
Rich, what do you have?
This is a pick thanks to Ed Niles. He sent me this article that's from the Washington Post, called "On a Bat's Wing and a Prayer." And it's a really nice article with really nice pictures about a group trying to discover more about the spread of, I just drew a blank. Marburg virus, yes. And so they've gone to a cave in Uganda, where actually, unfortunately, some people have caught Marburg, presumably from the bats, in the past, and trapped a bunch of bats and put radio GPS transmitters on them. And it's just a really nice story about this kind of field biology with, as I said, nice pictures. And actually, there are, as you scroll down to the bottom of the article, there are some links to related articles, and there's a related story about attempts to identify the host for monkeypox in the Congo Republic. So that's just as good. I like these field biology stories. They're great.
Yeah.
The cool thing about this is, well, first of all, they have the danger from getting the virus, but they also are wearing these waist-high, really thick waders that'll protect them from the snakes. The cobras and stuff.
Oh, right.
And also, one of the featured people is John Towner, who was a graduate student of Bert Semler's. So he got it started polio virus. And
yep.
Yeah.
I was thinking I met him when he was a student. Now he's 52 years old. Wow. We're just all moving on, aren't we?
We are. The time's arrow only points in one direction.
Oh, that's very good, Dickson. Where did you get that from?
That's a quote.
Anyway, it's really cool because they show putting these detecting devices onto the bats, like super gluing them on. And it's a great story. Thanks, Rich.
Nice.
Thanks, Ed.
Dickson Despommier, what do you have for us?
Well, I just watched this 2 nights ago. So this is my pick of the week, but it would have been my pick of the year, I think, because it shows you the life of a person who began much like ours did. In middle-class America, in this case, Chicago, and this is about James Watson, and went to the University of Chicago, and then ended up at Cambridge, collaborating with Crick. And it traced all of the controversial and non-controversial aspects of the discovery of DNA, and then what happened after that. And it's narrated, but it's also interviews, and it's also personal recollections. And to watch a person of this stature in the eyes of scientists, without knowing who he was, go from a fully respectable, believable, energetic person with a mission to explore as much as he could about a molecule, in this case DNA, to a failing, old, broken down, hate-filled, basically. I don't know any other way to put that. I think this is a must-watch for everybody. It's like watching a Theodore Dreiser novel unfold as a movie of someone's real life. Watson has the respect for what he discovered, and doesn't have the respect of any of his peers for what he's become. And what he's become is a belligerent, angry, self-centered, solipsistic person whose only point is that if he doesn't see the evidence, he doesn't believe it. And his main point that he's making now is that anyone from Africa that happens to be black at the same time, of course, hasn't got the same intellectual capacity as someone who's white. That's his big point. And he says there's no evidence to the contrary. And he keeps repeating this again and again. And all he's doing is basing this on the IQ testing, which is, of course, culture-driven and all kinds of flawed aspects. And it's a tragedy. It's an American tragedy. And that, by the way, was a novel by Theodore Dreiser as well. So you can actually watch this respectable scientist get deeper and deeper and deeper into the pit of his own prejudices. And by the way, Vincent, you'll notice that when we went to Cold Spring Harbor to do our live TWiV show, he wasn't there.
No, but a few years earlier, I went for a ceremony, and the ASM was putting a plaque designated Cold Spring Harbor as a milestone site. And there was a little ceremony which we filmed.
Right.
And Bruce Stillman made a few remarks, and Watson was there. In fact, I have a picture of him and Stillman up there with Stan Malloy. And they let him speak. And afterwards, Stillman said, I was totally scared about what he would say.
Absolutely.
But
He's
he behaved
unfiltered.
himself. He behaved himself, and because he said at one point that there's no use working on E. coli anymore. We're not going to learn anything. You know, stuff like that.
Right. We know enough about DNA now. Why do any more? He never said that, of course. But the point is that he shoots from the hip, and Kathy, you should. I don't know if you did watch it. Did you watch it?
No, I didn't. But Dickson, I have two comments. One is that a lot of what you describe, correlating with the time arrow pointing in one direction, just makes me think whether there's some neurological basis
could be
for this, the belligerence and so forth,
could be,
and so on. And the second is, what about all the genetic evidence that that there is no such thing as race?
I know.
I mean,
what? Of course.
No. So why hasn't somebody shown him that, or why won't he?
It doesn't matter.
He's fully aware of this. So Steve Goff, I just encountered Steve briefly today and asked him, how long into the past was Watson like this, and when did he start to change? When did he become such a complicated, ornery, and inappropriate person? And he said it happened a long time ago. I mean, it wasn't something that happened just recently because he's getting old. This is something that when he was in his 30s and early 40s, he was already saying outrageous things like this. So I think that, Kathy, you might be right. I mean, he may be aging more rapidly than, we have any right to judge him by. And therefore, a lot of this is happening against his will, so to speak. But every time he opens his mouth, something horrible comes out now. And he was on the Charlie Rose show, which was an interesting thing to watch in light of the fact that we now know what happened to Charlie Rose also. And Charlie Rose is sitting there, and he's absolutely yelling at Watson and saying, but you don't have any evidence for that. How can you possibly? And Watson's just sitting there as though he's not saying anything to him, and he just smiles at him back and says, well, I'm smarter than everybody else, and I happen to know these things. And that's his take on.
I didn't see it, but
or you'll be upset by it.
But someone said Joan Stites is
she's very, very heavily featured in it, and she tries to become Watson's apologist, and she fails because she admits to herself that, and in public too, and in saying it out loud, she says, I don't have an explanation for this. And there's a woman, her name is Morris, forget her first name. She's an administrator and a molecular biologist at Rockefeller University. She's also African American. And she was interviewed also in this. I don't want to spoil it for you because the impact of these people's statements on the statements that Watson was making are absolutely chilling. It's a fallen hero. Basically, it's a fallen hero. I mean,
I'm not sure. We have our listener pick has something related to this and sheds light on him a long time ago. I think he's always been a jerk.
No, being a jerk is one thing. Being totally inappropriate at every level and not being a scientist about it is another thing. That's another thing.
We know lots of scientists who, like
Well,
Peter Duesberg, who denies that HIV causes AIDS.
I mentioned George Wald as one of those other persons that went off on a toot after he got the Nobel Prize for Vision. I was at Rockefeller when Kiefer Hartline and he got the prize as a joint prize together. And Kiefer Hartline went on and kept working on horseshoe crab vision, and George Wald became a politician. And whatever he said, people had to accept because he has a Nobel Prize. That's bullshit.
I think this predated Watson's Nobel Prize.
It did. Yeah. And the other thing is, here's what I wonder also, and they didn't show anything about this, but they should have. What was Watson like between the time the paper was published in Nature and the time they got their Nobel Prize? That was a 9-year lapse. I wonder what was he doing during that time? And the answer probably is nothing. Nothing. And that's because he says,
I don't know about that.
What Watson says in this, again, a spoiler alert, not so much so. He says he doesn't do experiments. He doesn't go into the laboratory. He interprets other people's experiments. He has so much more fun doing that.
He stole Rosalind Franklin's.
No, it was clear. Carl Zimmer was very clear about this, and his take on it was that he didn't steal it. That, oh, come on, the other crystallographer, Maurice, come on, kids.
Maurice Wilkins.
Yeah. Maurice Wilkins gave it to him. Wilkins just thought he had a right to go into Rosalind Franklin's office and take whatever she had discovered and look at it and share it, and without her permission. And of course, that's an ethical consideration that's serious. And Sidney Brenner was on to talk about that aspect of it. And so every step of the way was fully explained. And this is one of the most remarkable hour-long sessions of a scientist's life that I've ever seen. And I was very sad for his family at the end of this. He has 2 children, and his wife is still alive.
Can I ask you,
sure,
should this have been done at all? Does he need more highlighting? Because this is PBS. A lot of people are going to see this. Is that good? Is it useful?
Very useful.
You think? Yeah.
Very useful. Very useful. I mean, someone who discovers that DNA is the molecule of life, basically, and then...
Well, he discovered the structure. He solved the structure.
With collaborators.
Yes.
But that's not what they said when they walked into the pub and announced that they had just discovered the secret of life. Yeah. That was what they said. That shows you the level of arrogance that they had.
So. He's one of the few scientists around who is a household name.
That's right.
Okay. Truly.
And so that makes
sure
a proper perspective on his life.
No question.
Relevant.
Yeah. No question.
Yeah, except that I think people look at these scientists. Look at them. Look at them.
Yeah.
Look how they behave. But I agree. I think you have to put it out there.
No one doesn't know that Watson isn't a scientist. He's a scientist. He is a scientist.
I mean, even Crick was highly critical.
Absolutely.
Highly critical.
Absolutely.
There's a pick about that in a moment.
Yeah. All right.
That's right.
Kathy, what do you have for us?
I picked something that was a paper I ran across yesterday when I was eating lunch. It's in Nature Methods, which I think is open access. And it includes somebody who is a co-author with me, a statistical geneticist, Alyssa Chesler, but several other people. And the title is Comparing Phenotypic Variation Between Inbred and Outbred Mice. And the premise is that people tend to use inbred mice because it's assumed that they have less trait variability. But they did an analysis in this paper, and they did not find that to be the case. And so it's a very short abstract. We conclude that contrary to conventional wisdom, outbred mice might be better subjects for most biomedical research. And they point out, for instance, that in the case of rats, people tend to use outbred rats, even though there's some inbred rat strains that are available. And they compared a whole bunch of different measures, and they have a lot of data sets. So I think it's statistically very strong.
Well, that's why a lot of people want to use collaborative cross mice, right? Because they're essentially
that's not even
it's not outbred, but it's better than inbred.
In fact, they compare what they use here as one of their controls is a different type of mouse, kind of like the collaborative cross mice, but they were developed at JAX, called the diversity outbred mice, and they use that as their outbred comparison. And so, but the gist of it is that that compared with inbred mice, defined outbred stocks are more appropriate and much more cost-effective research subjects in many biomedical research applications, except where precise genotypic regulation or standardization is required. For example, immunological, genetic, or molecular genetic applications. Right. So anyway, the point is that outbred mice are a lot cheaper too. And so if you are using inbred mice just because you think that they're not variable genetically across traits, that maybe you don't need to do that. I just thought it was interesting.
When I came here and started putting polio into mice, I used outbred Swiss Websters.
Did you?
Mm-hmm.
They're fine. Only recently we started using these horrible inbreds that eat each other and do all sorts of and don't breed well and
litter sizes. They're smaller.
Terrible. Yeah, ridiculously.
Yeah.
By the way, Nature Methods is not open access.
Cool. I got it on my telephone, so I thought that this article was.
That's cool. Thank you. All right, my pick is a New York Times article entitled "A Trump County Confronts the Administration Amid a Rash of Child Cancers" by Hiroko Tabuchi, just published January 2nd. These are about a group of kids in Johnson County, Indiana, who have been getting sick with weird cancers. And it's an old industrial site that was supposed to have been cleaned up years ago, and there are plumes of carcinogens underground that are releasing vapors into homes. And this was supposed to be cleaned up by the EPA. In fact, the Obama administration was supposed to clean it up, and it didn't get done. Now Trump has halted that, of course. And the people in this county who voted for Trump now are saying, hey, clean this up, help us. And, of course, it's not going to get done, so it's kind of a sad situation.
Where is Johnson County in Indiana?
Don't know.
I'll go look it up.
Why do you want to know?
Well, because I went to school there. I had a child there.
I think it was Franklin, Indiana.
Franklin?
I think when I looked at it yesterday, I thought it was Franklin.
Franklin, yeah.
Because there's a lot of coal mining there, too, of course. They have a lot of strip mines in Indiana. And, of course, the vice president is from, let's see, he's from Indiana. How about that?
Yeah, so it's very sad. You know, this...
So why doesn't the vice president get on this then?
This blind desire to save money and roll back restriction of industry, you know, it has consequences.
It's not a blind desire. It is a very deliberate...
of course it is.
Intentional thing to boost profits of industry.
It's blind to the consequences. Right?
Oh, yeah, absolutely.
Yeah. It's cost benefits in their favor.
We have... two picks from listeners. Anne writes a pick for your listeners if it hasn't already been highlighted. And this is a podcast with Peter Hotez in Texas Monthly. Peter is living in Texas, of course.
Yeah.
And it's about his book, of course. It's talking about similar things as we talked about on our discussion. So thank you, Anne. Happy New Year to all the Twix team members. Thank you. And then Islam Hussein sends a Twitter post from Tim Stearns, who is chair of Stanford Biology. Tim Stearns writes, in the interest of promoting an understanding of the history. Now, this has to do with the PBS show that you've just picked. Okay. This scathing letter from Crick to Watson in 1967 is a remarkable document. Crick lays out a detailed criticism of the draft of Watson's double helix, provisionally titled Honest Jim. And this, you can see the original letter, which is from Crick to Watson, and it says, "Dear Jim, the new version of Honest Jim is naturally a little better, but my basic objections to it remain the same. They are, 1, the book is not a history of the discovery of DNA as you claim in the preface. Right. Instead, it is a fragment of your autobiography which covers the period when you worked on DNA. Right. I don't see how anybody can seriously dispute this for the following reasons." Then he goes on. So Dr. Watson had issues way back in the 60s, maybe of different sorts or maybe of similar sorts, but kind of worth looking at.
His book was scathing personal attacks on all of his former friends and collaborators, all of them.
This letter is publicly accessible, by the way, and
Yes.
Yeah, it's amazing, and copied to Bragg, Wilson, Wilkins, Pauling, Wilson,
Yes.
Edsel, Doty, Kendrew, Perutz, and Klug.
Sure.
Wow.
All of them.
so this went to so Harvard University Press was going to publish the book.
They were.
So they sent this to the president, and he said no.
Right.
And then it was published by Atheneum, and the acquisitions person had previously been at Harvard Press. So I guess he decided.
Don't you have a copy of that on your shelf up over here?
Double Helix?
Yeah, sure.
I don't know.
I think I've seen it. But it's also one of the most popular books read by non-scientists about science.
This letter is just very,
It's very negative.
yeah, but a lot of errors.
Oh.
My objection, in short, is the widespread dissemination of a book which grossly invades my privacy, and I've yet to hear an argument which adequately excuses such a violation of friendship. If you publish your book now in the teeth of my opposition, history will condemn you for the reasons set out in this letter. So you can download the original letter. Amazing.
And he published it anyway.
Of course.
Yeah,
It
and
starts by saying
Of course.
that I've never met someone as self-centered as Crick. I think that's the first line of the book or something like that.
Yeah,
Right?
yeah.
No, he never knew him in a modest moment.
No, that's right. That's right. Yes. Gosh, a bunch of.
Yeah, and this speaks to Dickson's question: how long has
From the
he been a problem? And this is a 1967 letter.
That's right.
So Watson was off the rails a long time ago.
He was. I was at Notre Dame in 1965. In 1965, they invited all of the then alive people who had any role at all in the genetic code or discovering DNA, and they had like 8 or 9 people on the stage in front of the entire university. It was a remarkable symposium. And Watson sat there throughout the whole thing and stared at the ceiling and looked as though he wasn't even in the same room with these people. He just kept looking around like he was like in La La Land. And he was bizarre. I must say, just look at him. He was bizarre. So I feel badly for the image that he projects onto the public with regards to science. That's right.
Yeah.
Trust science, not scientists. Right?
Yeah. No, that's the deal. That is the deal.
So you can, in this tweet, there's a link to download the letter, which is really remarkable.
Yeah.
It had no effect whatsoever on him.
No, he wouldn't listen to anyone, I assume. All right, sorry. Go ahead.
You could put the link directly to that PDF. They don't have to necessarily look at the tweet.
Yeah, they don't.
Has anybody had a personal contact with him?
Mm-mm. I've seen him at meetings at Cold Spring Harbor. In the old days, when you went to a meeting, there was a reception at his house.
Right.
And he used to be wandering around
Right.
with drinking, and he was drunk often.
He was.
Yeah.
Sure he was.
But I haven't seen him lately.
Yeah.
How old is he? 80-something?
90.
90.
All right, that's TWiV 528, the first and not the last of 2019.
No, of course not.
You can find it at microbe.tv/TWiV. If you listen on your phone or tablet and you use an app, please subscribe to the podcast so we can know all your downloads. The numbers we got today are from downloads like that. We really appreciate them. Questions and comments should go to TWiV at microbe.tv, and please consider supporting us financially. We'd love your help to keep the show moving and keeping us moving. Microbe.tv/contribute. Today's show has been Dickson Despommier, thelivingriver.org, and Racaniello.org. Thank you, Dickson.
You're welcome, Vincent.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks, Vincent, and thanks for helping us do this for another year. It's been great. It's really been a lot of fun.
It's been amazing, in fact.
Yeah, it's a lot of fun, and nobody ever complains. I have to say, every week you guys do it, and that's way more than I should expect, right? So
we do it out of love for the field and for each other.
It's funny, on the other shows, like monthly, I say, when can we go to twice a month? No, I can't. I don't have time.
Look, when it comes to this podcast, I think we're like a viral capsid. You take one of us away and the whole thing falls apart.
That's a dominant negative, isn't it?
Yes.
Rich Condit is an emeritus professor at University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, always a good time. And I find, just to add my 2 cents, that as I get deeper into retirement, I'm bonded even stronger with this because it's my connection to that part of my life, and it's terrific to have it. It's great. So thank you.
You're welcome. Alan Dove is at turbidplaque.com, on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure, and I'm going to keep showing up to this until you tell me not to.
Oh, no, until I die.
Oh, okay.
Or I can't, if I start saying silly Watson-like things.
Right. No, we'll correct.
Then you fix me, tell me to shut up. I want to do this forever.
You don't have the genes for it.
The thing that I worry about is I'm not going to be at Columbia forever, right?
Nobody is.
And so I'm going to do this from home or wherever at some point. And is it going to be weird to be just sitting in a room doing it?
Doesn't bother me.
Surrounded by your friends on screens.
Yeah, but
welcome to the modern world.
Yeah, Rich.
Exactly.
Well, that's one of the best things. The thing that hurt me the most when I retired, though I did this quite deliberately, was that it cut me out of all of that social interaction. As you said, your whole premise for this podcast is we have these wonderful conversations in our offices. Let's publish them. Okay.
Sure.
And then you retire and that goes away. So that really hurts, but I've got the podcast. And so this is my conversation in the office. Okay. It's a virtual office. Yes. But it still exists. So it's not weird at all. It's great. In a weird kind of way, I look forward to gathering with you guys every week, and doing this. It's just great.
As Dickson said you said something which resonated, and I said, yeah, this brings joy to my life.
It does.
That's why I do it.
It does.
Over and over without worrying it.
Well, your joy is
Never
infectious. You've brought
joy
to our
this
lives.
Is the true meaning of bringing joy. Now I get it.
It's absolutely true. It's true. No, your whole demeanor changes when you sit down and you start to talk science.
Yeah, that's what you said last week. My demeanor changes.
It does.
Because there's a lot of surus here.
Well, no. And Rich, I want to add something to what you just said, because I'm retired too, and I've been retired longer than you had, and I didn't have the option of moving to where my grandchildren are because of my wife and her family. So we've stayed put in New Jersey. So I come into work every day. And the reason why, people say, what the hell are you doing that for? I said, the only difference between when I came into work before I retired and after I retired is they don't pay me anymore. But what do I get out of this? I come in here,
talk to people,
and it's an amazing world. My world didn't stop. It didn't stop. I mean, I'm listening. I don't understand everything, but I love being included into the conversation. That's basically what I love.
Yeah.
So that's why.
The conversations are everything. The camaraderie, smart people all over. Right?
Yeah, exactly.
It's great.
Right.
So I think you're right. The podcast will do that.
Yep.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASM for their support and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.